Good morning, everyone. Welcome to the PAG webinar series supported by Element Biosciences and Twist Bioscience. Thank you for taking the time out of your day this morning to join us, and thank you to our sponsors for your partnership. Today's topic is optimizing target enrichment for complex genomes, a case study in Southeastern blueberries. The question and answer portion of the webinar will be at the end. You can submit questions via the Q&A function. Any questions not addressed live will be answered via email, and we would love to hear your feedback and questions on this topic, so please don't be shy. And now I'd like to introduce our speakers. On today's webinar, we will have Kendall Lee, postdoctoral fellow, HudsonAlpha Institute for Biotechnology, and Raul Lodavia, senior product manager, Element Biosciences. Kendall Lee is from Atlanta, Georgia. Kendall earned her bachelor's in animal science with an emphasis on equine management from the University of Georgia. She earned her PhD from the University of Georgia and from the Institute of Plant Breeding, Genetics, and Genomics, where she researched the association between endophytes and tall fescue. She is now completing work for a USDA NIFA postdoctoral grant focusing on building resources for blueberry breeding in the Southeast. Welcome, Kendall. We also have Raul Lodavia. Raul is a senior product manager at Element Biosciences, where he manages new and existing products across the company's portfolio. Prior to Element, he held multiple roles in the genomics field, contributing to both R&D and commercial efforts for next-gen sequencing and multi-omics products and services. Raul holds an MS in biology and a BS in physiology and neuroscience from the University of California, San Diego, and is currently pursuing an MBA from the Haas School of Business at the University of California, Berkeley. All right, guys, well, let's get started, and I will now turn the presentation over to Raul. Thank you.
Thanks, Drew. Before we begin, I'd like to provide those of you who may not be familiar with Element just a brief overview. We were founded in 2017, and since then, Element has grown rapidly. We've raised over $670 million in funding and proudly hold a portfolio of over 500 patents and patent applications. In 2022, we launched our flagship benchtop NGS box, the AVITI, and in late 2024, the world's first and only fully integrated spatial multiomics box, the AVITI 24. Our products are now adopted in over 40 countries around the world. Additionally, Element has been an innovation powerhouse from its inception and has delivered a wide array of impactful product solutions year over year to address customer needs and continues to do so. In 2022, we launched the AVITI with the mission to redefine what's possible on a benchtop sequencer. Since then, we've delivered best-in-class sequencing quality, enabling smaller sample batches while maintaining reagent affordability. And we've also delivered several innovative solutions, our Trinity kits and our UltraQ sequencing kits being two of those. And with the platform, we have delivered unprecedented flexibility with dual independent flow cells, each with individually addressable lanes. With both AVITI and AVITI 24 sequencing, Element has delivered a simple, streamlined workflow. Users can load libraries prepared by their vendor of choice onto the sequencer. These libraries are then circularized before undergoing rolling circle amplification prior to sequencing. This rolling circle amplification produces no founder effect errors, a significant advantage compared to exponential amplification methods. Sequencing on Element instruments is powered by a foundational innovation, avidite-based chemistry, or ABC. An avidite is a dye-labeled polymer decorated with multiple copies of the same nucleotide. The multiple nucleotides on one avidite will bind to multiple active sites in one polyny, forming a very stable Velcro-like binding complex. And this allows for dramatically lower reagent concentrations compared to traditional SBS sequencing, and consequently lower sequencing costs while improving accuracy. To learn more about the sequencing technology, feel free to follow the QR code on this slide to our Nature Technology paper. So what do these chemistry improvements mean for users? Our researchers that are using the platform are seeing Q-scores in the mid-40s with our cloud-break chemistry, which you can see here in blue, and now into the 50s with our Ultra Q kits that you can see here in the red. And compared to other benchtop platforms, the AVITI shows clear improvements in read quality, index hopping, optical duplicates, and performance in homopolymer-rich regions of the genome. And these aspects of the sequencing chemistry affect nearly every NGS application. As I mentioned before, the system has also been designed and architected to maximize maximum run flexibility. The system has dual independent sides and independent flow cells, which allows users to perform 2 parallel runs with different parameters. Each of those flow cells are also equipped with individually addressable lanes, which allows users to use 2 different pools of libraries into the same flow cell. Now with AVITI 24, as I mentioned before, we have further expanded the capabilities on AVITI with certain software and hardware upgrades and integrated AI with greater processing power. We are now able to deliver not only our integrated library prep free single cell multiomics, but also NGS sequencing with up to 50% higher output compared to AVITI at the same reagent kit costs, which dramatically lowers the cost of sequencing for our users using AVITI 24. Element is delivering innovations across a wide variety of applications and spaces, including the agrogenomics space. With the low run costs, we've enabled the wider use of genotyping by sequencing and the flexibility to run any NGS application at a wide range of scales, all doing so very affordably. The way that the instrument has been architected and deployed allows for decentralization options to simplify logistics. And with our new chemistry, which I'll talk through in a bit, we've allowed for transformative workflow simplification for target capture using Trinity technology. And with that, I'd like to introduce a little bit about today's technology, Trinity Freestyle, which is Element's approach to improving hybrid capture workflows. So when we think about today's challenges with hybrid capture workflows, they're very long, laborious, have significant hands-on time, which really delays folks' sample to result timeframes. So when you think about the traditional hybrid capture workflow, you start with your DNA library and your probes of interest, perform your probe hybridization, and then you go through a bunch of B binding steps and washes before doing a post-hyb PCR, another QC, and then you finally load into the cartridge for sequencing. With our Trinity technology, we've cut out a lot of those middle steps, either by eliminating them completely or automating them onto the instrument. So now with Trinity, users can go from a DNA library using their target probes of interest, performing a 1-hour fast hybridization and loading that hybridized material into our sequencing cartridge, which then gets captured onto the flow cell directly. This technology has really revolutionized targeted sequencing by enabling a workflow, cutting hands-on time in half, and really accelerating time to results. We have built further on the capabilities of Trinity with our new product, Trinity Freestyle, that we have launched in conjunction with Twist Bioscience. Now with Trinity Freestyle, we've expanded the compatibility to the broader offering of Twist Library Prep portfolio, including their enzymatic fragmentation kit 2.0 and their FlexPrep ultra high throughput library prep kit, with more to come in the future. And with this Trinity Freestyle, we're delivering superior performance as we did on our original Trinity product, with quality of 90% greater than Q30, on target percent of greater than 85%, and mean target coverage of greater than 50x in the case of 2x150 with exomes. So Trinity seamlessly pairs validated library prep and capture panels from our launch partner with Twist with these new sequencing kits. So from Twist, users can order the library prep kit of choice, adapters and indices, the panel of choice, along with the hybridization reagents needed to enable the Trinity Freestyle workflow. And on the sequencing side, Element will provide the Trinity Freestyle sequencing kit and PHYX control, which comes with all the necessary materials needed to execute the Trinity workflow. With Trinity Freestyle, we've delivered significant advantages over the traditional workflows, reducing hands-on time, lower duplication rate with the elimination of that post-Hybe PCR, no wash buffer prep washes or conditioning, no heated washes, no need for any post-capture PCR or QC, and additionally, no need for any blocking oligos, significantly reducing that cost burden that blocking oligos provide. With Trinity Freestyle, we've really worked to enable sample to sequencer in just less than 5 hours. So compared to the traditional workflow that often has an overnight 16-hour Hybe, with Trinity Freestyle, that hybridization happens in as little as 1 hour, allowing for library prep and hybridization all to happen within just a few hours and data to be delivered as soon as the second day or third day, cutting hands-on time in half and enabling up to 2 times more sample processing capabilities. And with that, I'd like to pass it over to Kendall, who will talk through how she's leveraged Trinity Freestyle for her work in agrigenomics.
All right. Good afternoon. My name is Kendall Lee. I am a postdoctoral fellow here at HudsonAlpha Institute for Biotechnology in Huntsville, Alabama, in Dr. Josh Clevenger's lab. And today I am really excited to talk to you about how we have used this optimized targeted enrichment in Southeastern blueberries. So before I get fully going, a quick disclaimer. Element's products and services are for research use only. The opinions expressed in this presentation are mine only. So even though I live in Alabama now, I am a Georgia girl. As Drew mentioned, I'm from Atlanta, but I spent a lot of time in this little mountain town in North Georgia called Ellijay. And in Ellijay, we had a farm, and it looked like this. It's beautiful. Let's see, just clicker to go. Let me just run up. I'm sorry, we have a little difficulty here. Okay, there we go. All right, so that was the farm. I got to go visit quite often up in North Georgia. Fun fact, it's actually a winery now. So if anyone needs any recommendations for North Georgia wineries, let me go. The wine is actually pretty good. On this farm is actually where I developed a lot of my passions that I still have today. One of them being horseback riding. It's still my main hobby I have today, and this is me. This is a picture taken in 2000. This is me on a horse being very safe, as you can tell. No helmet, no shoes. This is my dad. But this is also where I had my first taste of blueberries. So on our farm, we had some blueberry bushes. They were wild, and there's also some cultivated. And anyone who has ever been to a U-pick farm or had some blueberries in their backyard knows that blueberries, or a single bush, can give you a lot of blueberries. So on this farm is where I developed my first taste of blueberries and my first love for blueberries. And in late July or early August is where I would first have these blueberries every year. And as much as we would try to have eat all of them fresh, we would end up having a lot frozen over the course of the year. And this led to a lot of treats. So we would have lots of muffins. If we were very ambitious, we would have frozen jams. And then my specialty would be blueberry cobbler. So today I'm going to be talking to you about, first, blueberry basics, so just a little bit about the background. And then we're going to be talking about blueberry tools, and this is where I'll get into some of the work we did with Trinity and Element Biosciences. And then I'm going to show you the results from this. So let's get into the background. So this is the work that I did with my USDA NIFA postdoctoral fellowship, which is in collaboration with UGA College of Agriculture. And our collaborator there is the blueberry breeder, Dr. Juliette Chu. So why blueberries? I think that obviously, why blueberries? They're one of the really unique things we have in this world where they're both tasty and healthy. Not only are they packed with antioxidants, particularly anthocyanins, but they are also super fibrous and they are full of vitamins. They are also super economically important. So blueberries are predicted to bring in over $3.7 billion in the market value for the United States in 2025. in honor of it being October, I thought the nice analogy would be $3.7 billion is actually double the market value for pet costumes in the entire pumpkin spice market. And everyone knows how important pumpkin spice is, so that's a big deal. The other super neat thing about blueberries is that it's one of the only crops that's actually native to the United States. And as you can see from this map of the top blueberry producing states in the United States, they're not really localized to one region. And part of that is because we have a lot of different types and species that are adapted to different regions of the US. And these are northern highbush varieties, southern highbush, and rabbit-eye. And what sets these apart? How are they adapted? What does that mean? A lot of this has to do with chilling hour requirements. And so in the colder states, we grow northern highbush. And these require around 1,000 hours of temperatures below 45 degrees Fahrenheit. So basically, that means they need about 41 days of temperatures where it is pretty dang cold. For me, this sounds kind of untenable. I mentioned I'm from Georgia. I live in Alabama now. That's about as far north as I think I'm going to make it. The southern highbush varieties need only about 200 to 300 hours. That's only about 8 to 12 days a year. So if you live in the Southeast, you know that is a lot more doable. And what chilling hours are is the hours required to break dormancy. So what do we grow here in the Southeast? So just honing in a little bit more on these Southeastern varieties of blueberries. The southern highbush varieties are tetraploids, is Vaccinium corymbosum. They are really good in fruit quality, so they're mostly what we market down here. They have very large berries, but they're a little bit of a smaller bush, and they're not quite as hardy. The southern highbush, on the other hand, I'm having a bit of trouble with my clicker. There we go. All right. The rabbit-eye, on the other hand, are a different species. They're Vaccinium aschii, and they are a hexaploid, just to complicate things further. They're actually native to the Southeast and just have been bred to have a little bit better fruit quality, and they're very low maintenance. So that's what I would suggest you plant in your backyard if you're looking for something. And blueberries have a really interesting history. So I mentioned they're native to the United States, but they haven't actually been bred for that long. Blueberry breeding has really only gotten started since about the early 1900s. And it started with the northern highbush varieties up in the Northeast. But people in the Southeast also wanted to be able to eat blueberries, and they wanted it on a different time frame. But that dormancy issue, that chilling hours issue, was a real problem. So the universities down here in the Southeast, some of these older universities of the University of Florida and University of Georgia started to start their own breeding programs in about the 1930s, 1940s. And University of Florida ultimately is the one who developed the southern highbush variety by crossing northern highbush cultivars with wild diploid lines they found in Florida. And they released the first southern highbush line in 1976. So let that sink in. That was less than 50 years ago we had our first southern highbush cultivar released. But we can't let University of Florida take all the glory. I am the alumni of University of Georgia, go Dogs. So we have to give credit to University of Georgia as well. Since their breeding program began in the 1940s, University of Georgia has released 35 southern highbush cultivars and 16 rabbit-eye cultivars. And I'm actually going to start a petition to rebrand Georgia as the blueberry state. And that's because the peaches are kind of losing popularity. In this bar chart you see here, from 2020 to 2021, we can see overall consumption across the United States of peaches versus blueberries, and peaches are just kind of losing popularity, whereas blueberries are really gaining popularity. And that is reflective in Georgia's market value, or how that's contributing to Georgia's growing and how it's contributing to Georgia's economy. So in the last poll on this in Georgia in 2021, blueberries actually contributed $350 million to Georgia's economy, whereas peaches contributed only about $85 million to Georgia's economy. So I think it's safe to say it's more of a blueberry state now than a peach state. So anyway, moving on, what are the traits of interest that we're after in blueberries? Well, probably everyone listening has eaten a blueberry, and you can guess what some of the fruit quality traits are. And there are dozens of traits that we could go after. But the ones that I am working with Dr. Juliet Chu to try to unravel some of the genetics on are days to flowering and days to fruit set, some phenological traits. And why are these interesting? Days to flowering is a really vital trait, actually, especially in the Southeast. So the way flowering works in blueberries is in the spring, they will go ahead and flower. And these flowers are ultimately what turns into the berries. But if we have a late freeze in March or so, and it'll kill those flowers, and then ultimately those bushes will not produce the blueberries. And then that's devastating, of course, to growers. And this is something that in real life, farmers have been battling. We also have issues, of course, with heat and humidity in the Southeast. And so the longer these fruits stay on the bush, the longer it takes to go from flowering to fruit set to actually picking these blueberries, the longer we have issues with fruit softening, skin ripping, and then issues with fungal rot, which then render these blueberries inedible. And then, of course, the growers cannot sell them. So our goal is to push back flowering time and push forward fruit set, shortening that flowering to fruit window. And that is what we're really trying to get after in my USDA NIFA project. And how does genetics come into play with all this? Blueberries are a difficult crop for breeders, especially when we don't have genetics at hand. Blueberries are a perennial crop that once you plant it in the ground, takes 5 to 6 years to become productive. And even if you want to start evaluating a fruit quality trait after planting it, you have to wait at least 3 years until you could even start collecting phenotype data on it. So let's say you're a breeder, you make a selection, you cross it, you have your new seedling that you plant, you have to wait a few years to even start getting phenotype data. But if you have a marker that is correlated with the trait of interest, you could go ahead and test these baby seedlings and start making early selections, speeding up that process drastically. So that's where we come in and trying to find markers that are correlated to these traits of interest. And the last thing I want to talk about in the background is actually the genetics of blueberries, because this really comes into play when we're talking about the tools that we need to use. We really need to think out what we're doing here. That's because the southern high-bush blueberries are autotetraploids. So they have 4 copies of the same genome. And here we can see the evidence of that with this cytological analysis, and you can see the tetravalent rings being pointed out here. And we have to consider dosage when we're thinking about the autotetraploids. So in a nice, lovely, simple diploid model, we can think about things as heterozygous and homozygous. So in a heterozygous, we have our A allele, B allele. That's nice and simple, heterozygous, AA, homozygous. But in an autotetraploid, we might have our homozygous AAAA, but then we start to have different doses of our alternative allele, where we could have 1 copy, 2 copies, 3 copies, etc. That is dose. And that is where we need to start thinking about the tools that we need to use going forward and how that might differ from some of the different crops and species that we work on. Okay. So now we can move on to the tools. Okay, so I've already said how our goal here is to find a marker, to be able to create a marker that is correlated with the trait of interest to speed up blueberry breeding. So broadly speaking, we need a few tools in our toolbox. We need a reference genome. We need our initial phenotype data so that we can make that correlation with our genotype data, and that's what we're going to spend a lot of this time talking about. And we need to do that analysis, of course. So currently there are some blueberry reference genomes out for diploid species and for northern highbush species, but there has not been a southern highbush or rabbit-eye. And as part of my USDA fellowship, I have built a southern highbush reference for Suzy Blue, which is a popular southern highbush cultivar from the University of Georgia. And I'm really excited about the Suzy Blue genome. Here you can see a contig map showing all 48 phase genomes. You can see the 12 haplogroups. The 12 haplogroups of the 4 homologous chromosomes. And at the end of each chromosome, there is a number. That's how many contigs are in that chromosome. And so this is a highly contiguous reference genome. And the red asterisks also indicate the telomeric motifs here, also indicating how complete this is. And the contig N50 is 12 MB, which is quite good for a Vaccinium reference genome. So going forward, I also wanted to make a primary haplotype for this. So what we did is pulled out the longest chromosome from each homologous chromosome group. And so that's what we use moving forward for the rest of the work I'll talk about in this talk. So moving on to targeted sequencing, the bulk of what we're talking about here. Targeted sequencing, just most of you probably know what this is, but for those who don't, targeted sequencing is DNA sequencing that it just focuses on a specific region of the DNA. It's a way to efficiently capture regions of interest without capturing what you don't want, where you can get high depth at a good cost. So for this, you need to have probes. And so here I'll tell you about our probe panel design. We use a 22K SNP panel that was designed by VatCap, which is a Vaccinium consortium group of the Blueberry community that they put together, as well as we targeted all the transcripts that are annotated for the northern highbush reference genome Draper. And so we ended up with this really awesome 148K probe panel. And I took all those probes and mapped them to my Suzy Blue primary haplotype. And here you can see the results of that, where every chromosome is pretty well covered, and it's all the genic spaces are covered, where the dips are in line with the centromeres. So we're really happy with that, and we're really set up to do the rest of this project. Next part is library prep. So we had our DNA already sent to us from Dr. Juliet Chu, so we needed to do library prep. And we partnered with Twist, who has this really wonderful library prep kit called FlexPrep. And FlexPrep is awesome for a number of reasons, but one reason is because it does early pooling, which cuts down on the plasticware, plastic consumables, and it also cuts down on how much you have to pipette and how much work you have to do. And the particularly amazing thing about FlexPrep is that it normalizes bioligation. So anyone who has done a lot of short-read sequencing knows you have to do normalization so that one sample doesn't eat up all your sequencing on the downstream processing. And you might have to do adding water, do all that kind of normalization that's very tedious and time consuming. With FlexPrep, you don't have to worry about that. So this is how we do all the libraries in our lab now. And then the way we worked with Element Biosciences to do this experiment is we took these FlexPrep libraries and we kept half of the library for ourselves at HudsonAlpha to do traditional targeted enrichment, and sent half to them to prep with their Trinity Freestyle. And that way, we had the exact same starting material. We also sent them the same exact probe panel, so everything was exactly the same going in. And that way, we could compare the workflow and the output. So you already saw some of this from Raul's presentation, but just to go over it briefly again. Here at HudsonAlpha, we did our full 16-hour hybridization and the temperature-controlled washes, yada, yada, day and a half of work. And then we sequenced it on our in-house NovaSeq. Over at Element Biosciences, I think they were having a much nicer time of things because it sounds like they only had to work for a few hours to get this really lovely data. And what did that look like? So in the end, we both got pretty good data, but the data that came out of the Trinity and AVITI was actually edged out the traditionally prepped data by a little bit. And they had, here you can see this scatter plot of the mean depth, sample depth versus the percent target covered. And you can see across the 192 samples that we sequenced, the overall mean depth was almost 50x, and the overall mean target site coverage was over 99%. So that's really phenomenal data and more than what we were targeting. One comparison I do want to show you between the traditionally prepped and the Trinity is the off-target data. And that is because the data that came off the Trinity and off the AVITI was a lot tighter. There was a lot less off-target site data as compared to the traditional that was sequenced on the NovaSeq. So overall, we were super pleased by the data we got. And as I'm about to show you with the results, that really phenomenal data had a pretty big impact on our results. So we'll get into that now. Reminder, we're looking at flowering time and fruiting time. And these are the tools we're trying to get. I showed you we have a good reference genome now for Southern Highbush, which I didn't mention, but all 192 samples that we sequenced are Southern Highbush. Phenotype data. I didn't talk about this yet, and I can't really take credit for this because this is the work of our collaborator, Dr. Juliet Chu down in South Georgia. But she and her teams have spent the last 3 years meticulously taking data on a large population for days to 50% flowering, days to 50% fruiting, and days to between flowering and fruiting. And I took that data and I just combined it with best linear unbiased estimate, which I think actually is, the acronym is BLUE, so Blueberry. And so now we have really good phenotype data going into this as well. So now we need genotype data, of course. This is a really big piece of this. And so we have really good sequencing data, of course. And just to give you a sense of the pipeline I used to get this data, for both the traditionally prepped data and the Trinity data, we took the FASTQs that we got off the sequencer and mapped it to Suzy Blue with BWA, and then called variants using Freebase, and then did some merging and filtering using BCF tools. So it was all pretty simple open source pipeline so far. But then we have to remember, it is an auto-tetraploid. So this is where things get a little bit different than maybe your typical crop pipeline. But luckily, there's a really awesome tool out here called GWAS Poly. This is made by Jeff Endelman. And this is where we can actually keep that dosage information. And this is what's really important for these auto-polyploids. Built inside this GWAS Poly software program, and it's an R program, is this function called VCF to dosage. So I'm able to take that merged VCF file and use this function VCF to dosage and get out my Geno file that keeps all that dosage information, and that's what I can feed into the actual GWAS. And something that I also think is kind of a feather in the Trinity's cap is that even after normalizing both the Trinity and traditional data down to the exact sample-to-sample yield, the Trinity still had about 18,000 more SNPs after all this normalization and conversion. So it retained more data for the downstream processing. And what we get here is across all the samples, we now have our genotype data retaining the dosage across all the samples. And so we're ready to actually get into the analysis. And what's really exciting about this and this kind of small sample size of 192, we're able to find 4 very significant SNPs for fruiting time and 2 for flowering time. And with GWAS Poly, we are able to look at these SNPs for a couple different ways under a couple different models, including under the genetic models of dominant and additive. And most of these were significant under the dominant model, but one was significant under additive. I just wanted to show you guys this because I think it was pretty interesting. The location, the significant location on chromosome 6, this is a location that is within a gene. Actually, all of these except for one were within a gene, which makes sense because we were targeting genes in our probe set. But this is a gene that is highly homologous to an F-box Celts protein. which has been found to have effect on flowering time in Arabidopsis. And even though this one had the effect on fruiting the other way than we want, it makes it later, I think that's still pretty cool and worth investigating. This other one, this location on chromosome 4, I just find pretty fascinating because it's like textbook example of additive genetic model. So that's all very interesting, but I also don't want to lose sight of what we're looking for here, right? We're trying to find something that pushes flowering time later and fruiting time earlier. And within these significant SNPs, can we find that? And the answer here is yes. So we are actually able to find meaningful markers. So here we have a site on chromosome 11 that when all four alleles are the alternative allele, we see that the effect size is 13 days of flowering time later. So it pushes flowering time on average 13 days later. So that's almost 2 weeks of protection from late freeze. So that's pretty significant. Similarly, we find a location on chromosome 7 where if only one of the alleles is dominant, we're finding an average effect of 17 days where fruiting is earlier. So 17 days of earlier fruiting. So that's more than 2 weeks that we're actually getting earlier fruiting. So we wanted to see among our samples of 192, do any of these have these positive allele combinations? So they have both this, these 4 alleles on chromosome 11 and this positive allele on chromosome 7. And then can we compare them to any sites that have pretty much the opposite of that, what we would call the negative allele combo, although it's not really negative because it's not showing that it's pushing back. It's just the opposite of the good, if that makes sense. And can we show this in a real world way? So we did find a handful of samples that had this what we call the positive allele combo versus the negative allele combo. And looking at the average of these samples, what we find is those with this negative allele combo on average would flower on March 4th and then fruit on May 18th. In contrast, those with the positive allele combo would flower on March 14th, so 10 days later, and then fruit on May 5th. So that's 13 days earlier. So what that all together, the negative allele combo are the ones that might be more normal. That's 76 days between flowering and fruiting. Those that have these positive allele combination only have 53 days between flowering and fruiting. So really, we're gaining, or maybe you could think of it as losing, 23 days that we are protecting our crop here. And right now, today, we could go ahead and design a marker for these 2 different sites and disseminate that to our collaborators who could then go ahead and start early selecting for blueberries right now. So to cover what we talked about today, just to recap, blueberries are delicious, they're healthy, they're economically valuable, but they also have a difficult genome to work with, and we need specialized tools. Thanks to companies like Element Bioscience and Twist, we now have tools at our disposal, and we have software programs like GWAS Poly that we're now actually able to decipher some of this work, or some of these difficult genomes, and are able to pull out significant locations that we can now go ahead and create markers that we can disseminate to blueberry breeders who can now select earlier in their program so we can continue to have better blueberries much faster. So with that, I'd like to thank you for your attention. Thank you for joining this webinar. I'd like to thank my mentor, Josh Clevenger, and Juliet Chu, my collaborator at the University of Georgia. I'd like to thank the entire Clevenger lab and also the co-lab, the Harcus lab, for really all the peers around me who helped me work through things like GWAS and reference genome assembly. It's really a team effort here. Thank you so much to Element Biosciences for their support with this. And thanks so much. We can move into questions.
Great. Thank you so much. We really appreciate it. Thank you, Kendall, for that great presentation. Thank you, Raul, for your great presentation. We will move into the question and answer portion. I don't see a lot of questions coming in yet, so please don't be shy. Please, we'd love to hear back from you and hear your questions, hear your feedback on how everything's going. So let me start by reading off the first question here. This question is kind of related to the high sequencing depth. What makes targeted sequencing a better option than skim sequencing for polyploids? And I'll just ask those, and Raul, you and Kendall can kind of decide who wants to answer.
Yeah, I can absolutely take that one. So with the dosage in mind, to be able to resolve dosage, you do need like a higher depth of sequencing to capture that. The higher the ploidy, of course, the more you need to understand the dose. And so skim-seq whole genome sequencing is not really a feasible option in terms of cost when you're thinking of needing 30x or 50x. So targeted sequencing kind of gives you the best of both worlds, where you can get high depth sequencing and actually just target regions of interest.
Great, thank you. Here's another question that just came in. I'll read this one first. Is there a yield penalty to shortening the flowering to fruiting time?
That's a great question. As far as I know, I don't think so. I would have to double check with our collaborator, but I don't think so.
Great. Thank you. All right, moving on to the next question. Are there other tools for running genome-wide association studies on polyploids that include dosage?
I'm sorry, can you repeat the first part of that again?
Sure. Are there other tools for running genome-wide association studies, GWAS, studies on polyploids that include dosage?
I hear you now. So as far as I'm aware, GWAS poly is the main one that really is built for polyploids. The current tools that you standardly use for diploids, like GAPIT or Tassel, you would have to diploidize the dosage, and in that way you'd lose some information. I think there's some other genotyping tools like PolyRad that you might be able to use, but GWAS poly is the best one to my knowledge.
Great, thank you. How do you increase the output from one flow cell between previous AVITI models and current models?
Yeah, I can definitely answer that question. So with AVITI 24 compared to AVITI, we've enabled certain software and hardware changes to the platform, which have allowed us to deploy improved analysis algorithms onto the system. And what this allows us to do is essentially extract more data from a given flow cell. And so users can use the same kit on AVITI 24 that they would use on AVITI and get up to 50% more output at the same price. So that dramatically lowers the cost for the sequencing.
There you go. Great. How would you move forward with marker-assisted selection?
Great question. Yes. So for marker-assisted selection, we would have to first create the marker, of course, and for dosage in mind, probably cast markers. I haven't actually done this yet, but I think cast markers would be the best way forward, or some sort of quantitative marker. And then you would go ahead and test your seedlings. As soon as you have enough tissue that you feel like you can take some, you would go ahead and test your seedlings, and that way you can say, this is a good one, this is a bad one, decide what you want to move forward with, and you can clear out the ones that you want to get rid of. And then you would still need to wait some amount of time for blueberries. They take about a couple years to flower. And then you could make your next round of crosses as soon as they flower, but you would not have to wait to do all the phenotyping work.
Thank you. Here's another question here. I'm just wondering, by inducing late flowering, will we miss out on yield and fruit ripening, or early fruiting leading to change in taste or flavor? He's just curious on this one.
No, it's a great question. I think that probably those are things that we'll need to explore some, but I can't imagine, I wouldn't think so, but I'm not so sure yet.
I know, right? Is it going to be a little bit more bitter? Is it, on the taste or flavor side of it? You wonder. Okay.
Yeah, you wonder.
In the plot with the in the genotypic classes, it would be expected 5 classes, but only 4 were displayed, 1 to 4. Do you have any explanation for that? I can repeat that if you need.
So in the genotypic classes, you mean where it's like all ref to all alt, right? I'm not sure which graph they're referring to, but in some of my alleles, there's some were just not present. Some didn't have every single class represented, if that's what they're referring to.
Sure. And Juliana, you can always come back with another question there and follow up on that if you'd like. That's who asked that question, so thought I'd throw that out there. All right, from James, did the original probe set come from Rapid Genomics?
I would have to go back to the 22K VATCAP. I can't remember off the top of my head from the paper where it came from, but I could get back to you on that. It's in the paper.
Sure. Thanks. And of course, we can always follow up on these questions via email. So if something didn't get answered, we can, get back to you in an email, no problem. Juliana, here's a follow-up question to her prior question. I was also wondering about the percentage of the phenotypic variation explained by the markers and the implication to use for marker-assisted selection.
So the effect size and some of those I showed for at least for the 2 markers that we showed with some of the variation, I think is what you mean by the variation, phenotypic variation, the 13 days for flowering time and then 16.8 days for the fruit time. And the implications for marker-assisted selection, hopefully would be that we would able to select for these idiotypes earlier on. But feel free to reach out to me, especially over email, if you want to discuss this a little further.
Yeah, and Kendall, you can always, we have Juliana's contact information as well, so somebody can get back to you, Juliana. You guys can have a conversation offline, no worries. All right, how many target enrichment probes are you using?
So in our panel, we had 148,000.
Wow, all right. And Heman, you can always respond to that if you have more questions to kind of expand on that if you'd like. Do you anticipate a mismatch between flowering and pollination if flowering is pushed back a few weeks?
That's a great question. So there's already such a wide, the flowering is already, we're seeing like a pretty wide range in the flowering time across a few weeks. So I am not so sure, but I wouldn't think so because it's already happening. These are plants that are already flowering at that time point. It's just how do we get more to flower at that time point?
Great. Here's one on the early side of it. How did you extract your DNA prior to the library prep?
Oh, great. Yeah. So actually, Dr. Juliet Chu's team sent us this DNA, and they extracted with CTAB. But in our lab, we use a wonderful kit through BioEcho. So pretty much most short read DNA extraction kits should work going into this workflow.
Great. We have another question came in. For your reference genome assembly with the OmniC scaffolding, did the genome fall into place easily, or was there a lot of manual curation involved? How was that?
Great question. It was not easy. There was a ton of manual curation, which we used JuiceBox to move it around. It's kind of an art form. And this is where I have to shout out some people in the Harcus lab. Zach Maharg particularly is really good with the polyploids. Spent a lot of time moving things around and just checking against reference assemblies and dot plots. And I can see why there is not more polyploid reference genomes out there. It was quite a long process to get this genome together.
Right? Wouldn't it be great if they just fell into place? Make it a lot easier for sure. All right. Well, it looks like that was the majority of the questions there. If you had a question that was related to cost for any reason and we didn't get to it, somebody from either HudsonAlpha or Element or Twist will get in touch with you via email. I think some of those questions are best answered in an email back and forth. But thank you so much, everybody. We really appreciate you taking the time and attending the webinar. We'd like to thank Element Biosciences. We'd like to thank Twist. We'd like to thank HudsonAlpha for your support and being here today. Look for the post-webinar survey via email for all of you attendees. Please take the webinar survey. We'd love to hear your feedback. We want to know, one, how we can do better. We want to know about other webinars that you're interested in hearing about so that we can go out and give you the best information that's out there. The on-demand webinar will also be available on the PAG website. Within a few days, we'll send you an email letting you know when that's ready. We look forward to your attendance at future PAG webinars and at PAG 33 coming up in January. We get to see all of you folks, which is going to be great because we get to see you in person. So we really look forward to it. We'll be having more talks on pangenome research, so it's going to be great at the live event. And one more time, just let us know what webinar topics you'd like to see. Thank you very much, and everybody have a great day.